Hi, I'm Sharon Neal. Welcome to this virtual poster describing our research. The work is inherently multidisciplinary, but measurement science is at its heart. We're developing tools for characterizing reaction networks in complex samples because so many important processes and applications require that we understand the dynamics of multiple interacting species. We use multi-channel optical spectroscopy and multivariate data analysis because this combination increases the selectivity of optical measurements, which are easily deployed for in situ monitoring, maximizing our ability to monitor multiple species simultaneously. When perfected, the tools we develop will be able to be applicable to reaction networks featuring many analyte types, but our current efforts target reaction networks featuring reactive oxygen species, or ROS, produced during oxygen photosensitization reactions as depicted in the simplified energy diagram on the left. ROS are interesting analytical targets because they play central roles in a range of biological, environmental, clinical, and industrial processes. In biological systems, ROS can even play competing roles, making accurate identification and quantification even more challenging. We're addressing this challenge from several vantage points: development of new probes, data enabled management strategies, and new data analysis tools. But we've barely scratched the surface of this broad area, and there are many opportunities for new students to craft projects emphasizing one or more of these uh, objectives. Stay tuned as students describe the rest of the poster, which highlights the instrumentation and some of the projects students are currently working on.
Hi everyone, my name is Claudia and I'm going to talk about the experimental setups we use to accomplish data collection in the lab. The images you see on my left show the three setups that we use most often in the Neal lab. The first, up here, is photodegradation reaction progress monitoring. This setup uses a broadband source, which is our xenon arc lamp, to irradiate reaction mixtures in a cuvette, which are then monitored by multi-channel spectroscopy. Another setup right here uses multi-channel broadband absorbance spectroscopy, which is taken on our lab's Agilent 8452 spectrophotometer for UV vis measurements. A cuvette containing the reaction mixture is placed in the instrument, and spectra are taken on the amount of light absorbed and transmitted by the sample. The last setup, down here, used most often in our lab, is multi-channel dynamic fluorescence spectroscopy, which uses our ISS K2 frequency domain spectrophotometer. The K2 can collect steady state and frequency domain decay measurements that we combine into Fourier transform emission decay matrices for analysis.
Hey guys, I'm Ina. My project is focusing on dynamic fluorescence measurements of Roux-Benguel photosensitization in octanol. In this investigation, I'm using wavelength-resolved multivariate frequency domain fluorescence measurements to investigate and monitor singlet oxygen photosensitization by Roux-Benguel in octanol, which is commonly used as a model of microheterogeneous medium in experimental and computational studies. Unraveling the spectrum response of reacting molecules that are distributed across the microdomains of microheterogeneous media requires specialized data analysis strategies. Multi-channel data acquisition facilitates data mining and mixture analysis. Matrix formatted frequency domain emission decay measurements are particularly amenable to numerical analysis, enabling isolation of the spectrum and decay profiles of complex sample components without a prior knowledge of the number or nature of their spectrum properties.
Hi, everyone. My name is Lakai Murray, and my project focuses on the investigation of photo-induced weathering of bare and surface passivated cadmium telluride zinc sulfide quantum dots using Fourier transform emission decay matrix measurements. Cadmium-containing luminescent quantum dots are being widely used because of their enhanced photophysical properties. However, their use of these toxic heavy metal nanomaterials has caused significant concern about the impact that cadmium-containing luminescent palm dots can have on natural systems and human health when they degrade, i.e. weather in the environment. Therefore, this study uses Fourier transform emission decay matrix measurements to investigate the wavelength dependence of the emission decay of bare and surface passivated cadmium telluride zinc sulfide palm dots and microheterogeneous media, ultimately to observe the changes in spectral properties and relative intensities in cadmium telluride zinc sulfide palm dot emission during forward-induced weathering, and to also investigate the RS formation and consumption during cadmium telluride zinc sulfide palm dot weathering.
Hi, I'm Deepak Sanap, a third year PhD student in Dr. Neal's research group. My work focuses on development of data analysis tools based on statistics and applied mathematics for the characterization of complex reaction networks. I collect and simulate optical spectra generated during oxygen photosensitization reaction to test the performance of the tools I develop. Here, characterization mainly refers to five broad steps. First, reaction progress monitoring using in situ multi-channel spectroscopic measurements. Second, selectivity enhancement by changing the measurement conditions. Third, perform multivariate data analysis to identify fingerprints of critical components of a reaction network. Fourth, estimation of kinetic parameters for critical networks of the species. Fifth and final, mechanistic analysis of reaction pathways to derive insight into it. These tools can have applicability in fingerprint extraction, spectral unmixing, pattern recognition, and reaction network analysis.
Hi everyone, Claudia again. My project involves using solid phase organic ionic materials as fluorescent nanoprobes for ROS. These materials, which are composed of mismatched asymmetric cations and anions, are gaining attention for their sensing capabilities and highly tunable properties. For my project, I plan on synthesizing these materials and evaluating them for their use as fluorescent probes for ROS. To accomplish this, I use multivariate dynamic fluorescence and time-dependent multi-channel UV-Vis measurements to monitor reaction progression, gather information about any spectral or structural changes that occur, and evaluate how well these probes capture and detect ROS. The diagram shown here visualizes an example of the reaction scheme and ion pair that I hope to use for my project.